and still few participants have not submitted their abstract. Okay, later I will announce the class roll number. Okay. Very good morning, Professor Jana. Professor Nikhil Ranjan Jana.
Yeah,
Very good
yes,
morning.
good morning.
Good morning. Good morning. I am Dr. Sanjeev Bhattacharya here, Associate Professor, UGC HRDC, University of North Bengal.
Uh-huh.
On behalf of UGC HRDC, I
Okay.
heartily welcome you in our refresher course, which is conducted in online mode. And we may start our program within two to
Yes.
3 minutes because there are 45 participants and they are joining one by one.
And most of them are from nearby college universities or
Yeah, exactly. There are total 45 participants and they are belonging to various colleges and universities of our country. Most of them are working in various colleges and universities of West Bengal and few are from outside Bengal. And they are discipline. They have various disciplines: physics, chemistry, electronic science,
okay.
computer science, and mathematics. So this is heterogeneous group, and it's our pleasure to have very renowned resource person, Professor Nikhil Ranjan Jana, Professor, uh, Center for Advanced Material, ICS. Actually, the thing is that actually I was in ICS during my PhD tenure up to 2008, and that time probably Professor D.D. Sharma came here to set up this group,
Yes.
this CAM group. I know I was present that time as a student, PhD student.
So the CAM is now changed. We have because of the UGC centers, so we have made some different restructuring of the departments. So
Okay,
now we have the School of Material Science that
okay, okay,
belongs to, so where I am. I belong to that school, and
okay,
most of the material science people's there with chemistry and
okay,
physics
okay,
backgrounds.
okay, okay. Sir, Brotoda is also there. Shomo Broto,
Yeah, he is
he is
another school that is called School of
okay,
Science.
okay, 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 okay,
That belongs to again
okay,
there are
okay,
physics, chemistry, biology people.
okay, okay, okay.
So your background, so when
Physics,
physics,
condensed matter physics.
and who was your professor here?
Professor Oshini Ghosh.
Okay, yeah.
38. We'll start our program from 10:30.
Okay, sure.
Okay. And dear teacher participants, who will give the brief description of Professor Nikhil Ranjan Jana?
Yes, sir. Myself, Dr. Manoranjan De.
Okay, okay, okay. You should be ready.
Okay, sir.
So we may start our first lecture session. It's our pleasure that we have very potential resource person, Professor Nikhil Ranjan Jana of ICS, Kolkata. So I would like to request our teacher participant, Dr. Manoranjan De, to give a brief description about Professor Jana. Over to you, Dr. Please.
Yes, sir. Good morning, sir. Good morning to all. I am Dr. Manoranjan De, Assistant Professor, Department of Mathematics, Mugbida Gangadhar Mahavidyalaya, Purba Mednipur, West Bengal. It is my great pleasure to introduce our speaker, Dr. Nikhil Ranjan Jana, in these two consecutive sessions. He is a professor of School of Materials, Indian Association for the Cultivation of Science, Kolkata. Professor Nikhil Ranjan Jana received his PhD in chemistry from Indian Institute of Technology, Kharagpur in 1994. He was a postdoctoral fellow at University of South Carolina from 1999 to 2001 with Professor Catherine J. Murphy, and at University of Arkansas in 2003 with Professor Jiang Gang Ping. He moved to Singapore in 2004 and worked as a scientist at the Institute of Bioengineering and Nanotechnology until he returned to India in 2008 and began his independent research career at Indian Association for the Cultivation of Science, IACS. He is currently senior professor at School of Material Science at IACS. His laboratory focuses on chemical synthesis of colloidal nanobioconjugate and their application as bioimaging nanoprobe and for prevention or curing of neurodegenerative disease. He has authored or co-authored more than 180 peer-reviewed articles for basic and applied science with a primary focus on tuning of nanoparticle surface chemistry towards various biomedical applications. He got Material Research Society of India MRSI medal in 2015. He was an editorial advisory board member of the journal SCS Applied Materials and Interfaces from 2016 to 2019. Also, he has been working as an associate editor of the Journal of ACS Applied Nanomaterials since 2019. So thank you very much, sir. With our armed regards, we welcome Dr. Jana, sir, to take this online platform. Over to you, sir.
Okay, thank you, Professor Dev. So, and thank you, the University, North Bengal University, for selecting me. And I have about 100 slides for two lectures. And there are some slides are there are so much technical things, but don't worry about it because of the wide audience of these people who are from different backgrounds. I have tried to summarize some of the covers all of the aspects of the nanobiotechnology, what people are doing around the world, and I will just briefly outline most of them. And there are many technical things. If you don't understand, forget about it. And I try to give some sense of the subject and the areas that we cover. Okay, so I'm going to the present. Can you see the slides?
Yes, sir.
Yes, sir.
Okay. Okay.
Yes, sir.
Okay. So the title of my presentation is it any times if there is a problem of seeing slides or my voice is okay, is it?
Yes, sir.
Yes,
Okay.
sir.
If any problem, let me know at any time. Okay. So the title of my presentation is biomedical application of nanoparticles. So you heard about the nanoparticle, the term, and you might heard, some of you might know that there are a lot of applications in biology, and we will try to cover some of these aspects, how these nanoparticles are important in the context of understanding biological processes. So these are the four topics we will cover. Again, they will be briefly covered. The first topic I will try to elaborate a little bit more, because most of you should know this subject, the role of nanometer length scale in biology. The second area is I will try to briefly cover some of the synthetic methods, how we can make nanoparticles. Of course, you need to understand some of the chemistry. I will just highlight some of them. Third one is approaches of making nanobioconjugates. Because just making nanoparticle is not enough. You have to transform them into some conjugates, means linking with some biomolecules, so that they can be actually useful for different applications, biomedical applications. And finally, I will try to cover what kind of applications we can do using these nanoparticles. What is nanoscience? The science at the nanometer length scale, controlling matter at typically in the range of 1 to 100 nanometer scale. Controlling matters, you have to control, you have to fabricate, you have to design. Fabrication of device, can we make device at this length scale using some of these nanomaterials? And we know that nanometer means 10 to the power minus 9 meter. But how small it is? We have some idea about 1 meter. If we compare nanometer with respect to 1 meter, it's very difficult to imagine for us because it's so small. We have some idea about Earth. If we can compare a small football or a marble with respect to Earth, we can actually compare how small is 1 nanometer with respect to 1 meter. And so small means, are they smaller than molecules? No. Roughly, one glucose molecule, glucose you might have, most of you know, a glucose molecule is roughly 1 nanometer in size. And if you are a chemist, you know that in glucose molecule there are lots of bonds. Bonds between carbon and carbon, bond between carbon, hydrogen. And these bonds chemists used to fabricate, make, do a lot of modifications of these kind of molecules. So these nanoparticles are actually larger than molecules. So typical size of a molecule is generally less than 1 nanometer, unless they are very large macromolecules. And size of a protein, DNA, virus, they are about 1 to 100 nanometer. What is the size of coronavirus? Roughly 50 nanometers. What is the size of a cell? Living cells, these are microns, more than 1000 nanometers, 10 to 100 microns, micrometers. This is a comparative scales. In the nanometer scale, if you put different items, a glucose molecule, as I said, it's about 1 nanometer. Antibody, water molecule, it's 1/10 of nanometer. Antibody, 10 nanometer. Virus, roughly nanometer, 10 to 100 somewhere. Bacteria, a little bit larger, 10 times larger. A cancer cell or just living cells, microns, 1000 nanometers or more. Now, important question comes: why nano size is interesting? Why it is important? It's important because it is an intermediate between atomic or molecular state with respect to bulk state. Bulk means like water or a metallic piece. There are lots of atoms or molecules are present. But if you divide them into small and small and small, when you go to nanometer range, it is actually intermediate between a molecular atomic form versus bulk range. Means if you decrease the size, if you come in this range, there might be some interesting features. What are those? Size dependent properties. Just by changing size, you can expect, in many cases, you can expect some changes in physical or chemical properties. That's why it becomes important. High surface area, that's very much known. Most of the people who do not understand the subject, they will only say the nano is important because high surface area. You make into divide into small pieces, surface area will increase. That's the common sense. That's well known, but that's not the only aspect. Size dependent property and reactivity. Reactivity will be different. For example, the one simple example, aluminium. It's a metal. It's a bulk metal. It's not very reactive. You can keep it in the atmosphere for a few days, few years, without much of destruction, so without much of reaction with oxygen. But if you break them into small size, nanometer size, they become explosive. They react vigorously with atmospheric oxygen. This kind of chemical reactivity will be significantly different in this small size range. The subject area where we are interested and we will cover today's talk is last one. It's linked to last one. The biomolecule or cellular objects are in this length scale. Why they are in this length scale? What is the advantage? Why nature selected so many small objects, nano-sized objects, nano-sized components? What are their unique features, roles that are useful for biological systems? So these are the areas for which nanoscience are important. Here I showed the nanoscience in the context of whole history of science. The microscope, you see the microscope is invented around 1600, through which we can see small objects that we cannot see under using our eye. Naked eye cannot see it. So microscope can see something smaller. What smaller? You can see something roughly high-end microscope. You can see micron size like bacteria and some cells, cellular features. You can see using microscope. Where actually we use light for diffraction, refractions, and based on that, we get an image of a samples which are very small. We magnify and see them. And then photography and imaging, that was developed when? 1800, 1900. That was developed first in France. And the photography principle, if you see the latent image making, and nowadays digital cameras are different, but the earlier photography where we use, what is that called, negatives, then development of these negatives, where we use a small silver clusters, small nano, these are actually nanoparticles. So people did not know earlier, but actually they know that it was very small. And so in that respect, we were aware of these small particles and their importance in photography. And most important one, Faraday. Michael Faraday, we know he was linked to electricity generators, but actually he first prepared colloidal gold nanoparticles, but he termed them colloidal gold salts, and he were aware that these unique colors of these gold nanoparticles are coming from small size, and these small size interact differently with the light, sunlight or visible light. And that's how they get different colors. And without knowing these colors, how they are generated, ancient peoples use these for decoration of glasses, windows, using colloidal gold particles. So Michael Faraday developed a method of making gold colloids, and that is still preserved in Royal Society Museum. They are so stable. But these are all without seeing them. As a chemist, we know that we cannot see atoms or molecules. We cannot see atoms or molecules, but we can deal with them easily without seeing them, because we have many other instruments through which we can indirectly see the bonds, break the bonds, make new bonds like NMR, etc., etc., mass spectroscopy. But seeing is most powerful approach if we can see. So electron microscope was developed 1900, 2000, sometimes 1940, 30s, 40s. Electron, instead of using electron microscope, we use electron beam to image some things. And because the wavelength of electrons are much smaller than light, we can actually see something in nanometer length scales. So these developments help the subject nanoscience grow in this current forms. And this is the electron microscope, where a typical, don't have to go to the details, but if you have seen it, this is the type of microscope, where actually we have to image samples under very high vacuum. That's the limitations, but that's a very powerful approach, because we can directly see what we are making. We can see things of something less than 100 nanometer under this microscope. In addition to, so in order to develop, or if you want to do research, you need to have these electron microscopes. In addition to that, we also have some other high-end microscope, atomic force microscope, scanning tunneling microscope, where we use a cantilever, a tip, sharp tip, and that tip sense something surface features, and based on this sensing, you can translate into an image of a surface, image of surface features in nanometer length scales. So using these advanced instrumentation, we can actually see molecules. We can image molecules. These are some of the examples. We can manipulate molecules and matters at these length scales, nanometer length scales. And this is another interesting story. You can see right side. This is small nanoparticle, semiconductor nanoparticle. They have short name, quantum dots. What you see, when you image them in the single nanoparticle form, each of these nanoparticles are blinking. This fluorescence is blinking. It's like stars. Every nanoparticles, they blink. But if you see them in solutions, the left side, different size solutions, you don't see this blinking. This is an average property. So when you see, when we look at the average property, we miss the individual level actual properties are actually they are blinking. Their fluorescence is not continuous. They are discrete. The left sides are actually same material, quantum dot cadmium selenide. Just by changing size, you can get all these different emissions just by varying size. And each particles they have blinking property. Fluorescence are blinking. So, in order to understand the material property, you have to go down to single particle level. You have to image, you have to collect spectroscopic features at the single particle level. And by doing average property, you might miss many of the things. So these are typical history of nanoscience, where Michael Faraday first prepared gold salts. Richard Zsigmondy, which is the time scale 1865 to 1929. He first developed synthetic methods of making colloidal gold. And that time there was no electron microscope, so he used ultra microscope, where light scattering is used to detect size of a particle. Scatter light with the size, the small particles can scatter light. That scatter light is analyzed to indirectly determine the size. Langmuir-Blodgett films, monolayer of molecules you can make using like oil in water interface. If you put a drop of oil in the water, they will spread into a monolayer of oil molecules. It's a monolayer. Two-dimensional molecular layers. That's actually nanoscience. The two dimensions they are nanometer scale. Richard Feynman's famous book. We can do plenty of things if we go down to nanometer scales. Electron microscopes developed between 1930 to 50. Cluster science for a chemist. Chemistry subjects, you know that small molecule clusters, atomic clusters, few atoms, few 10 to 100 atoms clusters. By changing the size of these clusters, we can make a lot of difference in their properties. Nanoscience is highly interdisciplinary. It involves many different branches of science. Supramolecular chemistry, colloidal cluster science, material science, engineering science, applied physics, because in this dimension there will be new laws. Most of the conventional laws may not work. You have to redefine. And molecular biology, why? Because many of the objects are nanometer, length scale, macromolecules, their property, behavior. If we want to understand, if we need complete understanding of the nanoscience. And why at this time scale? Why we are thinking at this time? Why at this time? The reason we are in a position to deal nanometer length scale. We talked about electron microscope. They are invented just 10 years back. And chemical science, atomic molecular science, polymer science are developed. And in chemical science, we have a problem. We now know what are the problems of making large molecules. If you want to make polymers, a large molecule, we always end up with a size distribution, molecular weight distribution. If you want to make a polymer with a single molecular weight, it's a problem. Very synthetic limitations. If you want to modify a small functional group of a large molecule, there are interference from other functional groups. There are lots of problems. If we want to deal, we have to deal nanoscience. And in addition to the physics, chemistry, material science, there are a lot of discoveries of instrumentations. And if we want to design fabricate something at we can go up to micron size, but going nanometer size, there are a lot of difficulties. And for that, we are in a position to deal, and we need to make at this length scale to advance the subject for advanced understanding of the subjects. And what are the current researches that we do? These are some of the areas. For a chemist, synthetic you have to make materials like there are chemical companies who sells a lot of small molecules. People who cannot make it, you can purchase it. People who cannot make it, who like to make some fancy materials, they can purchase some starting materials and then build up his own. So similarly, in nanoscience, we have to develop a library of nanoparticles of different sizes, say like rods, wires, different quality of materials. Then only with this availability we can make variety of functional materials, and we can do a lot of applications, medical science, chemistry, energy, communications, consumer goods, etc. So in nanobiotechnology, what we do, we actually make materials, try to understand nanomaterials, their size dependent properties, and we try to see what nanoscale objects are present in biological systems and what is their functional role. What are the functional role of different nanoscale units present in living cells or life processes? And we combine these two subjects and then design nanomaterials so that we can use them for different biomedical applications. On the right side, some of the examples, what kind of biomedical applications we can do. Detecting single biomolecules. There are lots of biomolecules who are linked to different disease, early stage disease diagnosis. So detecting single biomolecule. Tracking single molecular events inside a living cell. Bioimaging followed by renal clearance of nanoprobes. Suppose we have a tumor in a body. We have to image where it is. For imaging, we need most cases, we need contrast agent. We have to put in the body from outside. But for that, they might stay in the body. That's very bad for our body. So they might stay in our body. So ideally, what we want is that they should be clear from the body after imaging. So we have to develop those things. Bypassing endosomal lysosomal degradation of nano drugs. Whenever we use drugs, they have a lot of misuse. Means they are only a small fraction of it are actually used. Rest of them needs a lot of side effects, and they are degraded by our body system cells. So how can we minimize those things? Can we design nano drugs? Direct cell membrane translocation of drugs. These are some of the advanced features we need for advanced nano drugs. Wireless therapy. Suppose you have something in your body, very remote areas. Can we do some wireless therapy without surgery? Brain delivery of drugs using ultrasound, sound, light. This can be used without surgery. Can we do therapy? So some hints. So here this slide actually shows a list of examples. Don't have to go to the technical details. I will cover some of them in the later part of the talk. but these are least. Lot of in the biological system, lot of nanostructures, components present in our body systems, living systems. Biomembrane, for example, cell membrane, for example. What is the depth or the thickness of the membrane? 5-6 nanometer. Lipid wrapped. These are actually some assembled organelles in the cell membrane through which cell sends from outside surface. Clutching vesicle, when some things are cell took from outside, they trap them inside and they made of proteins. These are called clutching vesicle. And see all length scale, micro nanocytes, microtubules. Microtubules, and these are kind of highways present inside cells through which something enter from outside to inside and goes different parts of the cellular components. They are made of proteins, microtubules, nanocytes, length and diameters, micron size length. Iron oxide, nanoparticles, some of the bacteria use for something. I'll go into details later. Iron oxide bacteria, diatom. Diatom is a body structures of green plants. Amyloid fibrils, which are linked to Alzheimer, Huntington disease. These are protein fibrils, nanometer length, in the micron size, nanometer diameters. Lotus microstructures. Because of these microstructures, they are water repellent. Ferritin structures, some of the proteins, iron-based proteins. COVID, the green one, this COVID-19, the coronavirus sites. Kidney stone, bone microstructure, malaria pigment, they are all examples of nano-size materials, units, or the components, biological components. And these are the synthetic nanoparticles. Actually, chemists, physicists, primary chemists, they make these materials. These materials are made in our laboratory, just examples. These are all electron microscopic images. And each of these samples, you see the scale bars and the materials, iron oxide of different sizes, manganese oxide, gold, silver, cobalt oxide, and you can also see, although most of them are spherical, some are anisotropic, rod-like, some other cubic shapes. So all these we can make. In high resolution, again under electron microscope, you can also see some more additional features, plane defects, atomic defects, that in high-end electron microscope you can also see those features. And those defects are sometimes linked with the physical, chemical, catalytic and other properties, optical properties. So we can correlate them. This is a beautiful, well-known examples, size-dependent property of materials. 3 examples I have, 3 materials: silver, gold, and cadmium selenide. Silver, by changing their size, particularly shape, all in nanosize, typically between 10 to 100 nanometers, micron size wares. These are all colloidal solution of these particles. You can see their colors. You can vary the colors from whole visible to NIR regions. Similarly, for gold, just by changing their size or shape, nanorods of different length, plates, you can get different colors. For quantum dot, cadmium selenide, this is the colloidal solution of this cadmium selenide under UV exposure. you have to shine with UV light, then you can see their fluorescence. And these fluorescence, their colors depends on the size. These 6 vials, the size roughly 1, 2, 3, 4, 5, 6 nanometers. So 1 nanometer to 6 nanometer, if you vary the size, you can get 6 different colors. And these emissions, right hand side emission spectra, they are very narrow compared to any conventional organic dyes, as you know. So these quantum dots, the emission colors are size dependent, emissions are narrow, and in addition to that, you can excite with UV light. We excite all of them. All the 6 samples you can excite with a single light. And single blue laser can be used to excite all. Single UV laser can be used to excite all. These are the advantages, practical advantages during our biological imaging and application where we will use them as a fluorescent levels. We have to understand size effect when we want to use them in biomedical application. Size is important. Size, surface chemistry. Here is actually mostly we are showing the size. If you want a renal clearance, means something you want to image, inject in the body, image, do some function, and then everything should be clear from the body. For that, you need size very small, less than 10 nanometers. If you want photo or magnetotherapy, magnetic field based or light based therapy application of nanoparticles, better is small, larger size is better for therapy, but smaller is better for clearance from body, clearance from cell. So depending on the requirement, you have to design. EPR effect, what is that? Enhanced permeation retention effect. That's linked to tumors. Let's say if you have a tumor in a body, you want to target tumors. Typical size for targeting, if you want to target the physical barriers of these tumors as leaky blood vessels. So if you want to use that, size should be 50 to 100, 200 nanometers. If you want to cell targeting cells, size should be between 10 to 50 nanometers because endocytic uptake requires that size. If they are too large, cell cannot do it, cannot take it. Again, from the chemistry and physics point of view, increased smaller size means increased surface area, increased surface curvature, increased surface to volume ratio. That may lead to interaction, enhanced interaction with the surface through the surface molecules, increased interaction with bulk phase, increased colloidal stability, so many aspects we have to consider. And for that, size is important. So now I will go through few slides which are more details of the role of nanometer length scale in biology. This is the structure of diatom, microscopic structure of diatom. What is diatom? Diatom is the green plant present in sea, and most of our oxygen supplies in our earth is because of these diatoms that present in sea. And what is their surface? Their silica based cell wall. And you see the scales, the micron scale, and you see the features inside. It's about nanometers, 2 nanometers. And inside cell, there are a lot of components, macromolecules, proteins, components, cellular components. For example, mitochondria. Mitochondria is micron size. There are hundreds and thousands of mitochondria in each cells. And each mitochondria, there are membranes, and then there are different sub-mitochondrial components. They are nanocytes. Golgi apparatus, then nucleus, nucleolus, then cellular functions that are linked to cavules and other components. These are ribosomes, endosome, lysosomes. These are all less than micron size, nano-size components present in biology, in cellular cell, inside every cells. Eating and drinking activity inside cells, in a cells. So in the bottom, you can see, depending on the nature of particles, size of particles, cell eat them via different uptake process. They have different names for biological people. They know it: phagocytosis, macropinocytosis, clathrin, caveole endocytosis, etc. And you see the sizes. If they are very large, macropinocytosis, phagocytosis. If their size is between 50 to 100 nanometer, clathrin, caveole endocytosis. And in the top one, you can see processes, uptake processes. And in a cell, there are highways and motors, nanomotors, that drives something from outside to inside by catching them and passing them from one side to others. Just a second. Hello? 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 I thought you people are calling for some issues. That's right. Okay, so there are a lot of highways and diways, and nanomotors inside cells, and through which something from outside goes inside cells and drives towards nucleus or some other cellular components via these highways. These highways are made of proteins, and their lengths are microns, but diameters are typically 25 nanometers. So nanoparticles as unit of crystal growth. We used to think that we used to explain most of the things based on atomic and molecular concepts. If we understand property of atoms or molecules, we can explain bulk properties. But nature sometimes use nanoparticle as a unit to make bulk materials. And actually there are advantages. We can actually make, if we want to make defect-free bulk materials, nanoparticle unit is much better compared to molecules or atoms. There are hundreds of reasons. And here you can see a bacteria that navigate in a sea using magnetic nanoparticles. They can align magnetic nanoparticles using their proteins in the body along or opposite to the Earth's magnetic field. So they use Earth's magnetic field and alignment of their magnetic nanoparticles inside body. By doing that, they navigate in a sea. So nanoparticles, magnetic nanoparticles, they use as a unit. Bone consists of nanoscale weak bonds. Our bone is made of inorganic and organic. Organic is actually collagen. Inorganic is calcium salt, calcium phosphate salt, called hydroxyapatite. And this hydroxyapatite is actually brittle. It's a powder. And collagen, organic, very soft. But you can make bone, which is very hard. It's not brittle. How is it possible? It's possible via nanoscale intimate interaction. Interaction between inorganic nanoparticles, hydroxyapatite plate, nanoplatelets, with interaction with collagen fibrils, fibrillar organic materials via multiple weak interactions, weak bonds. These bonds are called sacrificial bonds. Why sacrificial? Because under pressure, some of these bonds can break, sacrifice. And by doing that, actually they can relax a little bit, but they don't break easily. And these are reversible bonds. Once you release the pressure, those bonds will again, the broken bonds will again connect. So these are reversible but weak bonds and many bonds. And these many number of bonds are possible because of nanoscale interaction between inorganic and organic components. Catch bond in a bacteria. Bacteria use a proteins through which they attach to the surface. And the flowing, if the solvent or the dispersed media is under flow condition, they use their proteins, they can sense the flow of a fluid, and this sense will be used to catch the surface. That's why it's called catch bond. So you can remove this bacteria. How can you remove this bacteria from some fruits? You cannot use a flowing water. Flowing water means this bacteria will firmly attach on the surface of your fruit. If you want to remove it, just dip it into water for some time and then remove it. If you dip it in non-flowing water, they will be detached. If you use flowing water, they will be more firmly attached. So use a catch bond. In the bonding is in the nanoscales. The nature of bonding, if you want to understand, you need to go down to nanoscale. Shape matters in biology. If you make a spherical versus wire-like shape, wire is better for targeting, for delivery. Let's say your drug is inside a sphere or wire. Which one is better? Wire is better because wire can align along the flow of the blood. So if you inject these wires in the blood, they will stay in blood for a long time by aligning them along the blood flow. And longer time in blood will help to target tumors or some other organs. So for the similar nano size, wire is better than spheres or other size. And biological systems are highly crowded with a lot of molecules. And this crowding limits their movements. But they like freedoms. But in the crowding environment, their freedom will be lost. But they somehow want to increase their freedom. How can they do it? With a small volumes, they align. But the alignment means they will lose some random movement. But actually they can increase their freedom of rotation. Rotational movement they can increase. So they increase different kind of freedom. And this rotational freedom enhancement they can do And actually, this is only possible under crowded environment via aligning them. Align means different kind of liquid crystalline structures, nematic, smectic, or columnar or rings. So rod-like particles, if it is very narrow, so these are synthetic materials. These materials are all are made in my lab sometimes, different time scales. So you see increasing aspect ratio, length to width ratio. If it is small rods, they align in symmetric orally. If they are plate-like structures, columnar orally. If there is a mixture of different length of rods, they will be nematic orally. And if very uniform rods, little longer, larger, there will be again symmetric. But they're very long, wire-like structures. They used to make bundle structures. You might have bundles, nanotube, carbon nanotube bundles. The nanotubes of carbons are made in large scale, macro scales, industry scales, and their bundle problems are very common. So these assemblies, nanoscale assemblies, these are also important. You have to understand because in cellular environment, these are very common. This is more details of the cell uptake mechanisms where you can see, depending on the size and surface chemistry, cell can take nanoparticles of different size and shape by different approaches. And different approach of taking means they are trafficking into different parts of the cells. Their processing biochemistry will be different depending on the surface chemistry. We heard about different neurodegenerative diseases like Alzheimer's, Parkinson, Huntington's. These are all linked to nanoscale aggregates of defective proteins, mostly in neuron cells and in brains. So in order to understand the progress of the disease, origin of the disease, we have to understand nanoscale features, what is happening. These are the protein aggregates that are linked to different neurodegenerative diseases. These are electron microscopic image of the proteins, protein fibrils, different kind of fibrils. All are linked to Alzheimer's, Huntington's disease. So I hope you understand the importance of nanoscale features, nanoscale units present in biology. Now I will quickly go through some of the synthetic methods. It's very brief. 4 or 5 nanoparticles, how you make it. So for a chemist, it's very well-known approach is start with the metal, some of the molecular precursors. If you want to make metal nanoparticle, for example, start with the metal salt. Gold nanoparticle, if you want to make, use gold salt, gold chloride. And then, because gold is in oxidized form, gold plus 3, you have to use some reducing agent that will transform Au plus 3 to Au0. Just a second. Hello? Number 1? কোন জানার কাজে এসছে মানে কিছু আমি হ্যাঁ বুঝতে পেরেছি আমি এখন বলতে পারবো না এখন মিটিং এ আছি বুঝেছো পরে আসতে পারবো So for making gold nanoparticle you start with gold salt at a reducing agent that transform gold ion to gold atom, and then gold atoms will aggregate into clusters and nanoparticles, like as I showed in the right side, and then it will form nanoparticles, large clusters, nanoparticles, and these nanoparticles have to be stabilized with a surfactant or polymers. So polymer or molecules stabilizer at the surface of these particles, you will get a colloidal nanoparticles. Making high quality nanoparticles, if we want to make high quality means different size, specific size, surface chemistry is well defined. If you want to make them, these are some of the approaches. If you want to make small particles, short growth time, nucleation and growth. Nucleate into small particles, then grow into larger size. If you use short growth time, you will end up with a small particle. But if you grow for longer time with more additional precursors, you will end up with large particles. If you use fast nucleation and short growth time, that's the ideal to make small particles. Slow nucleation and fast growth. Only few nucleation center in a solution. You start with a metal solution, only few nucleation sites, and grow in those nucleation sites for a long time, you will end up with a large particles. But in the same solution, if you add a reducing agent, very strong one, so that so many nucleation centers are formed, and you don't have much amount of precursors also, all will be exhausted very soon. Then growth will be very short. So you will end up with a small particles. So small particle versus large particles you can make by these approaches. And the middle one is seeding growth. You first make a small seed, grow it into larger size in presence of precursors. And you can stop at different size depending on what size you want, depending on how much precursor you added. And the last one, if you somehow cannot make a single size or shape, you end up with a mixtures. You can separate, but separation has a lot of limitations. But this is one of the approaches. This is here one approach, depletion attraction. So just concentrate the mixture of rod and spheres. If you concentrate, because the rods have assembly property, they precipitate, and you can use this approach for isolate rods and spheres, sphere and rods mixtures. So those are the approaches, general approaches. This is one specific examples. I think many of you who work with these nanoscience, they might have heard or made it. This approach is actually available in internet. You can see the video of these making materials. So you start with a gold salt solution, heat it. Actually, two reagents you need. One is reducing agent, trisodium salt. And it acts as reducing at high boiling point, high boiling water. And another reagent is gold precursor, gold salt. So in a boiling gold salt, you add trisodium salt. And at the initial stage, the color is a little bit dark violet, and with time, particles are actually attaining the correct size. It's roughly 10 nanometers. Colloidal gold. You know, the method was developed long before by Richard Sigmonti, and we still use it. Little bit modification, perfections. It's a beautiful particles you can make. And by varying the amount of trisodium citrate, you can control size from 10 nanometers to 50 nanometers. So gold nanoparticles, colloidal gold. This is quantum dot, cadmium selenide. How will you make it? Again, the precursor is what? For the cadmium, there are two precursors, cadmium precursor, selenium precursor. Uh, So cadmium precursor, first you dissolve cadmium some salt in a solvent under some solvent which you need to heat little bit 60-80 degree, and then the solvent is a high boiling solvent. You can heat up to 250 to 300 degrees centigrade. This is a moon. This is called hydrocarbon. Hydrocarbon high carbon, long carbon chain hydrocarbon. This is called Hrdc, and you can heat up to boiling point in 320. So you can boil up to heat up to 300 degree. So only cadmium precursor is there. At this high temperature, you inject selenium precursor. Inject under rigorous stirring condition. Magnetic stirrer is there. Under stirring condition, you are injecting. Why the injection? Because rapid mixing. The nucleation growth is very fast. So injection, and after injection, we have to reaction starts. Selenium will react with cadmium. Selenium precursor, actually there are some selenium precursor or cadmium precursor, they actually degrade and convert into cadmium unit and selenium atomic units. They combine to make cadmium selenide clusters, nanoparticles, aggregations. And surfactants are there. You see the hydrocarbon surfactant, hydrocarbon based surfactant, they cap the surface. So all is happening. Nucleation growth, quantum dot formation is happening at high temperatures within second, few second to minute. We have to stop the reaction at 10 seconds. 10 seconds. In order to get violet color, violet emission. If we can stop at let's say 30 seconds, you will get green emission. 1 minute, you will get red emission. In between, you can get other colors. How can you stop the reaction? You take a mixture, a small part of mixture in a solvent, cold solvent, or whole solution can be cooled into room temperature by injecting another cold solvent, another, let's say, octadeciane, you are injecting into, rapidly injecting them. So stop the reaction. So reaction, nucleation growth at high temperature, and suddenly stopping the reaction at certain time, at few seconds or within a minute. So when you stop, that will dictate your product, whether you will get. And if you are immature, you will get completely non-fluorescent material because they will grow into very large size, more than 10 nanometers, micron size. You will end up with non-fluorescent materials. So making these materials, you need expertise. You need to learn from lab. For example, in India, hardly 20 or 30 research groups make these high quality nanoparticles. Why we can make it? Because we are trained again from advanced laboratories in other countries, US or other countries. It's very specialized synthetic skill unit. And people who are trained in our laboratories, they can again make. If you just read the literature and try to make it, very difficult to make. Making gold nanoparticles. So this one, we made gold nanoparticles 10 nanometers. If you want to make smaller than 10, this is another very beautiful, powerful approach. What is that? It's a thiol-based surfactant. So we are not telling how we make it. First, make dodecan thiol gap gold nanoparticles, which are very small, 1 to 5 nanometers. Very similar process. Instead of water, we use organic solvent-based approach, where gold nanoparticles are kept with some surfactant, BT is dodecan surfactant kept particles. These are 1 to 3 nanometers via some brushed method. I am not going through details. Another standard methods. You can make gold nanoparticles in organic phase, aqueous organic interface, and finally transform into organic phase. So gold nanoparticles are dispersed in organic phase, toluene, let's say, and you are heating it. This is called austral deepening. You are heating it for at certain temperature and at certain time. You will get different size. 1.5 nanometer is converted into 3.4, 5.4, 6.8 to 9.7. So you can change the size, increase the size from 1.5 to 10 nanometers. But each case is a very narrow size distribution just by heating. By heating, what happens? At high temperature, smaller particles dissolve and grow into larger particles. And at a certain size, depending on the temperature, will control what size it will stop. Time, it will dictate what size it will stop. So it's called austral ripening, sometimes it's called digestive ripening. It's a very powerful approach of making. And these kind of materials are now commercially available. You can also purchase them. These are all solid particles. If you are to make hollow particles, air inside, nothing inside, how can you make it? Of course, there might be some pores, but how can you make it? It's called galvanic replacement or Kirkandel effect based replacement. What is that called? So, if you, let's say you made a silver particles, and this is the one silver particle, and add gold salt. Gold will deposit outside, and silver will leach in the form of silver ion. This is a galvanic replacement reaction because in the electrochemical series, gold is more noble. Gold, there is no other reagents. Gold ion is converted into gold zero, and it takes electrons from silver, and silver forms silver ion. Silver ions, once they form silver ion, they leach. And why it makes hollow? Because in nano size, reaction is very vigorous. And silver moves its place. Silver moves. Moves means it moves from inside to outside in order to react with gold ions that are present outside. So that's how hollow comes, hollow shape is formed. That's called Kirkandel effect. Vigorous reaction leads to place exchange. Place change. Silver changes its place from inside to outside. And in outside, because it becomes silver ion, it's leach out into the solvent. And instead of silver, gold is deposited at the outside. That's how hollow structures form. This is a principle. It can be adapted to make many kinds of hollow particles. Iron oxide. If you want to make iron oxide nanoparticle, any metal oxide, this is a common principle. We start with metal salt. This salt is actually carboxylic acid salt. Instead of common carboxylic acid, it's a fatty acid, long chain hydrocarbon based fatty acid salt. If you heat in organic solvent, you can get oxide materials. Early stage, they are anisotropic. With time, they increase into size. Depending on what size you want, you can change the reaction environment, temperature, and the acid that you used, their length, long chain, that will dictate the size, and the temperature will dictate the size. If you want to change, you want to make different shapes, These are the example. This is one of the approach. If you want to make gold nanorods, first make a small spherical seed, then use some surfactant that attach at a certain side face of a growing particles, so that the other faces will grow. This surfactant is called a specific surfactant CTAB, cetyl trimethyl ammonium bromide. It absorbs at the surface, side face, and the rod-like structure will grow. Because end faces, the surfactants cannot absorb properly. The gold will grow, gold will deposit at the two ends. Seeding growth, we can also transform them into non-seeding growth. So these are the type of gold rods shapes you can control by adjusting reaction environment, spheroids, length, width, long rods also you can make, you can separate. There are different ways of making size control. If you want to make different size, there are two approaches. Either core particles, core inorganic particle you can grow and then cap with a molecular layer of other molecule, other polymers, or you can use left side, you can use same size core particle but coat with a cell, increase the cell thickness like polymers or silica. You can increase the silica cell thickness. By that way, you can also control the size from 10 nanometer to 100 nanometers. So two ways of controlling size. I'm just showed some approaches. I think that's all about the synthetic methods. Most of them, what I showed is via chemical approach. Why chemical approach is most powerful? Because diverse ways of making particles, diverse ways of controlling size and shapes. We can have other approaches like top down. You grind them into large macro scale materials into small size. There are also limitations. Very difficult to control size in 1 to 100 nanometer length scales. And most of the unique size dependent properties are in this length scale. So chemistry plays major role in making these materials. And here in these slides, just showed some of the particles which have very specific functions. And next lectures I will show mostly these particles. So the first one, you see fluorescent particles. They have fluorescence. If you excite them with some light, blue, green, or UV, they fluoresce. So these particles can be used as an imaging probe, fluorescent probe, like fluorescent dyes. What are the advantages? I will give in the next lectures. So nanofluorescent nanocarbon. See the length scale, the scale bars, all is in nanometers. Calcium phosphate, it's a hydroxyapatite, bone materials. You see that they are anisotropic. Why they are important? These materials can be used, they are very non-toxic, or the body has, so this can be used as a carrier, delivery carrier, drug delivery carrier. They have another interesting process. They are piezoelectric. If you put pressure, there can be some electrochemical reaction at that surface that can be used for different applications. Light-induced ROS means reactive oxygen species generation. These are TiO2. TiO2, NF doped, nitrogen and fluorine doped TiO2. So it's not conventional TiO2. Conventional TiO2 are white color. These are bluish-green colors because they have very small percent of titanium +3. NF doping leads to these situations, and they can capture visible light. TiO2 cannot capture visible light, but this NF.TiO2 can capture visible light and can produce this, do some photochemical reaction, and can be used for degradation of toxic materials, can be used for degradation of cells. BaTiO3 nanorods, sound induced reaction. You can use ultrasound to generate reactive oxygen species. Nanovitamins, these are organic nanoparticles with a significant 10 weight percent of vitamin C is present. Vitamin C delivery into cell. If you can deliver vitamin C in high dose, they can be a source of hydrogen peroxide and do some induce oxidative stress in the cell. Polyaspartic acid. Aspartic acid is an amino acid. Aspartic acid is polymer micelle as a delivery carrier. And last three, nano green tea. Green tea means, green tea has some component. Green tea has a biochemical component that's called EGCG. Full name, that's called, it's a kind of polyphenol. It's a polyphenol. So green tea has a very specific polyphenol. This polyphenol has an antioxidant property. If you can make their nanoform, they can be delivered into cell in much better way. So that's why nano green tea. Nano quercetin, another polyphenol. Nano trehalose. Trehalose is a carbohydrate. So these trehalose nanoform, they have very specific unique applications in treatment of neurodegenerative diseases. So all these are made in our labs. Some of them we made on our own approach. Some of them are adapted from literatures. So all these have specific functions. We use them for different applications. So maybe I should stop here and start the next session, or I can continue. I do not know. What do you want?
Sir, 10 minutes break, sir.
10 minutes or 15 minutes?
Okay.
Okay, 15 minutes. Then we will come back after at around 12.
Yes, sir. Yes.
Around 12. Okay, we'll see you again.
Okay. Good morning to all dear participants. This is the time for short break. So one small announcement. Can you hear me?
Yes, sir.
Yes, sir. Okay. Regarding your seminar presentation, so 12 minutes will be allocated for presentation and 3 to 4 minutes for question answer session. Okay. This is the answer of your first query. The time duration 12 minutes for presentation. You may present in terms of PPT or chalk and talk, or you may deliver your lecture verbally. No issue. There is no compulsion to use PPT or some supporting tools. Okay, you may describe your presentation verbally, as well as you may use chalk and talk method or something like this. Okay, there is no compulsion. 12 minutes is the time limit, 3 minutes for question answer session. So since it is seminar presentation, our adjudicator's decision will be final. He or she will ask some questions, some queries related to your presentation. Your presentation should be lucid and should be interdisciplinary, so that your friends from other discipline can understand little bit from your presentation. Okay, and adjudicator may ask some questions, or he may allow to ask questions by the participants. Okay, that will be entirely dependent on the decision of the respected adjudicators or judges. Any other? Second thing, just a minute. Roll number 2. Probably roll number 2. Some participants have not submitted their seminar abstract. Just a minute. Class roll number 4, are you here? Class roll number 4?
Yes, sir.
Okay, you have not submitted your abstract.
Yes, sir. Yes, sir.
Why?
Hello.
Why you have not submitted your abstract?
Hello. Yes, sir. I have submitted 14 November.
But I have not received your abstract. Please send it once again. Please send it right now. Send it once again. Class roll number 4.
Okay, sir. I have submitted. Thank you. ठीक है.
Okay. Class roll number 36. Are you here? 36. Okay, class roll number 36. You have not 36. Saroj Mondol. Dr. Saroj Mondol, are you here? You have logged in, but you may not present over here. Dr. Saroj Mondol, can you hear me? So I request anybody from the audience, please try to connect with Dr. Saroj Mondol in this regard. He has not submitted seminar abstract.
Sir.
Yeah.
Sir, please check it.
No, it is not received.
Sir, could you please check also the spam folder if probably if it has gone there? It sometimes happens like that.
No. It is not in the spam. Regularly, I have checked the spam folder. No issue. It is not here. It is not. Some Manobindu has sent the abstract to HRDC, not HRDC4, but I have collected from HRDC. All the email IDs are accessible. No issue. But it is not received.
Okay.
It is not received. And 36, Saroj Mondol, you have not submitted your abstract.
Yes, sir. I will send tomorrow, today.
No, by today, by right now, not today.
Oh, okay, sir.
Right now, 14 was the deadline. By today.
Yes, sir. Yes, sir.
4, 5, 6. Okay. Other people have submitted. Roll number 4, I have not received the abstract from roll number 4 and roll number 36. Okay. Okay. If you will have any queries, I will discuss it later. Okay. I will discuss it later. Okay, classroom number 4, if you have any problem, you may call me up personally over phone. Okay.
Okay, sir.
Okay, classroom number 4, Devikanandan Maji.
Yes, sir.
It should be HRDC 4, not HRDC PROG 4. Previously, we used that email ID, but right now it is HRDC 4. You may check your email.
Okay.
Okay, HRDC
Okay,
4,
sir.
not HRDC Pro 4.
Okay, sir.
Dr. Jana, your screen is shared. Please, would you please turn it off? Because you are probably writing some email or something.
Okay, Dr. Jana, hello? Can you hear me?
Actually, he put off his headphone, so he
Right.
couldn't hear our voice.
Okay. Anyhow, I try to connect.
Sir, if any message is given to his chat box, he may observe it.
Issue. He is typing just sending one mail, no issue.
Yes, sir.
Sir, please check it.
received. Thank you.
Okay, thank you, sir.
Now it is received.
Sir, that mail was FIP08 program.
Right.
স্যার নেক্সট ইয়ার কি স্যার অফলাইনে এই প্রোগ্রামগুলো হওয়ার সম্ভাবনা আছে স্যার?
We are trying to conduct it in not only fully offline, maybe in blended mode. Participants may come over here and some resource person will deliver their lectures physically and some may deliver in online mode. Okay. In blend, probably it will be conducted in blended mode.
ওকে স্যার।
So welcome back.
ওয়েলকাম স্যার।
Hello.
Welcome, sir.
Welcome, sir.
I hope all of you are joined again.
Yes, sir.
ইয়েস স্যার।
Okay, so the roadmap for the applications. So far we discussed about nanoparticle synthesis, about the importance of various nanoscale units in biology. And now I will quickly go through applications. If we want to apply these nanoparticles for specific biomedical applications, what we need is synthesis, then some kind of surface processing, then some conjugation chemistry. What is that? Linking with some chemical or biochemical to the surface of nanoparticles so that they can be selective to certain components of the cells or certain biomolecules. And then application. Application may be for imaging, for detections. So why you need it? You cannot directly link nanoparticle with biomolecules. You need some chemistry, chemical linking, physical adsorptions. Even for that also, we need to do some processing. What kind of processing? This nanoparticle may be charged so that electrostatic interaction can be used for assembling some molecules at the surface. Some functional groups so that we can do chemistry for linking with molecules. Why? So why these are important? Because so what we need is actually colloidal nanoparticles, and their surface should have some chemical or biochemicals. All these nanoparticles should have some chemicals or biomolecules at the surface. And cells or nanoparticles can interact with this biological interface through these surface molecules. Biological interface, for example, cell cannot directly see what is inside nanoparticles. They can only see or face. They can face what is there at the surface. And colloidal nanoparticles are important because if they are dispersed, they are accessible by all sorts of biological environment. If they are insoluble, they may be nano size, but if they are insoluble, if they are not dispersible, they cannot move around. They cannot move inside cells. They cannot move inside body. And for that, dispersibility is important. So dispersible or soluble and specific chemistry at the surface. So that's why this term is important, nanobio conjugate. And for that we need to know, so far whatever nanoparticle we made, what are their types? They are type 1, type 2. Type 1 is the early stage. Somehow we make particles with some surfactant or polymers at the surface. Surface is not very much well defined. Type 2 is a new generation. It started from 1990. So these are actually surface of self-assembled monolayers, surfactants monolayers. They are processable. These nanoparticles are a little bit processable. They don't break easily. They don't aggregate easily. You can do some precipitation, redispersion, processing. Type 3, these core nanoparticles are trapped in a network of polymers or some inorganic materials, cross-linked cell. So these are more robust. You can process, you can do conjugation chemistry, so many things. And these are final generation, new generation, type 3 and new generation materials. We like to make, if you want to use them, ideally you should make type 3, third generation particles from the first or second generation. These are the processes. What are the processes? Silica coating around the nanoparticle surface. Polyacrylate coating around the particle surface. Polymer coating around the nanoparticle surface. And these are the precursors of, if you want to make silica coating, you need to use some silane precursors. They are all available, commercially available. And if you have a chemist, you can quickly select them. And other polymers, sometimes some polymers are commercially available and researchers made them, so you can use those polymers. So once you make it, this cross-linked cell What are the advantages? Better protection of nanoparticle, high colloidal stability, introduces functional groups, chemical groups, so that we can do conjugation chemistry, linking chemistry. And right side two images are shown that nanoparticle can be precipitated. How? By changing the dispersion medium or the changing pH or adding salt can precipitate. And this precipitate does not mean that nanoparticles are destroyed. They are not converted into bulk particles. Each core nanoparticles they are still retained the structures because they are kept with cross-linked cell. And that's why this precipitation is reversible. You can collect the precipitate, disperse in a fresh media, right media where they are soluble, they again original properties they will be dispersed. Quantum dots are commercially available with these kind of structures. Core cadmium selenide, then protection with zinc selenide and other inorganic layers to protect the fluorescence, and then polyacrylate polymer cell, and then some biomolecules. And in addition to that, you can link your own biomolecule that you like. For that, you have to buy polymer coated particle, and they will provide some procedures how to conjugate with your biomolecules. Why do we need to conjugate nanoparticle with different molecules? For example, here different functional groups, and then why? Amines, carboxylate, olide, these are all chemical, or polyethylene, these are all chemical functional groups. Chemists usually do it. Why? For doing some linking, for making them charged, making them lipophilic. Polyethylene glycol for making non-ionic surface so that it won't interact with cells or anything. That will also sometimes important so that it reduces non-specific bindings. The most important is the last one, linking with peptide, protein, oligo, carbohydrate for targeting. If we want to target a specific cell components, specific organs, then you have to link with specific biomolecules. So once you make nanoparticle with primary amine, for example, at the surface, which are shown in the middle, these nanoparticles can be transformed into different functional nanoparticles. Folate terminated nanoparticle, biotin terminated nanoparticle, galactose terminated nanoparticle, trihalose, phenylalanine, triphenylphosphine, peptide, peptide. So now we can make nanoparticle terminated with specific biomolecules. Folate biotin. Why? These are vitamins. These vitamin receptors are overexpressed in certain cells, cancer cells. So we can target cancer cells, those cancer cells using these nanoparticles. Target and image those cancer cells. If you have quantum dots, you can use fluorescence property of quantum dot to image. We can also do right side some more fancy conjugation chemistry by changing the number of biomolecule attached to nanoparticles. This number change will lead to change of interactions with the cell surface. Changed interaction will lead to different uptake mechanism, cell uptake mechanism. So that's why they are important. You can also start with a amino acid. For example, here in the middle you see aspartic acid is shown. Aspartic acid is converted into polyaspartimide, then converted into different sorts of polymers, different polymer micelles for different applications. From these polyaspartic acid will converted into nano-quercetin, nano-green tea, nano-arginine, and it can be used as a coating material for nanoparticles. So all these are, you need a lot of chemistry. Once you did these things, so these are some of the examples of these nanoparticles. So the top lines are showing the nanoparticles for purpose. Design nanoparticle to bypass endocytosis. Endocytosis is cellular uptake process, trafficking process. You can bypass it by this kind of surface chemistry, arginine terminated nanoparticles. Design nanoparticle to control endocytosis. This, see, different functional groups are there. This is again design nanoparticle as a delivery carrier, delivery of proteins or vitamins. Then design nanoparticle to target different subcellular compartments. Something it should target inside cells, mitochondria or lipid droplets inside cells. Again, these are again nano green tea, nano trehalose, nano glutamine, nano trehalose drug, polymer based. So these are all designed for inhibiting protein aggregation inside cells, inside intra or extracellular matrix, because this Alzheimer Huntington's, it can happen inside cells or outside cells. So ideally you have to minimize this aggregation process. Also you have to remove these aggregated toxic proteins from cells, from extracellular space, from body outside. So you have to design nanoparticles that can clear this toxic protein aggregation. So these are some of the nanoparticles. So once we have these particles, now I will quickly go through some of the applications. What applications? Imaging, molecular microscopy imaging, small animal imaging, biosensors. These are some of the areas where these nanoparticles, the design nanoparticles can be used. If you want for detection purpose, this is a very general approach. Here the nanoparticle you see, the nanoparticle is functional nanoparticle. Nanoparticle with antibody, terminated with antibody. This can be used for detection of a protein. So this, the protein that we want to detect, it has some specific antibody that we can make nanoparticle functional with that antibody. So once this protein binds with this antibody, means binds with nanoparticle, because nanoparticle has a color, fluorescence or just normal colors, we can see the protein. So proteins are dropped, proteins are spotted in a tissue paper or in a membrane, cellulose membranes with specific fine membranes. So this protein spot can be visible. Otherwise, proteins are colorless, you cannot see it. But if your nanoparticle will help this protein to see. Protein spots can be seen by using these functional nanoparticles and the color of these nanoparticles. We can do more fancy detections, more specific and more sensitive detection by further processes. This is another example how to detect oligonucleotide using gold nanoparticles. So the top one is the oligonucleotide with the blue border lines. That oligonucleotide you want to detect. So we know the complementary sequence, and that divided into two parts, 1 and 2. and those are 1 and 2 are linked with two different set of gold nanoparticles. So we have two different reagents of gold nanoparticles attached with 1 and 2 oligonucleotide. Once your target nucleotide is mixed with two sets of gold reagents, gold colloid color will be changed because of its complementary sequence, they interact with each other, and that interaction will lead to particle-particle aggregation. Gold nanoparticle aggregation will lead to color change. This color change can be used for detection of your oligonucleotide. This is a very powerful approach. We can detect single oligonucleotide mismatch. Surface and Raman spectroscopy, single molecule detections. Gold and silver nanoparticles are very powerful materials. They have surface plasmons. Their colors are due to surface plasmons. Plasmonic electron clouds. These plasmons can be excited by lasers. And any molecules that are absorbed at the surface of these nanoparticles, they will experience more electromagnetic field. So Raman signals will get enhanced. Billion-fold enhancement of this Raman signal that will lead to single molecule sensitivity. So detection of a single molecule, highly sensitive detection can be done using gold or silver nanoparticles. There are a lot of, this subject is very mature, very intense study. This needs several lectures. I'm just giving a brief overview. So graphene, you heard this. This is a functional graphene. Colloidal graphene linked with peptide, linked with a organic fluorescent dye via a peptide-based linkage. This peptide can be cleaved by enzyme, chymotrypsin. Because this organic dye is very near to the graphene, and graphene can quench the fluorescence of this dye. The fluorescence is off. Once its enzyme is present, enzyme can cleave this peptide bond. Organic dye is away from this graphene, and this will lead to fluorescent switch on. So enzyme can be detected. Similar approach, cholesterol detection. Here, cyclodextrin. Graphene is conjugated with cyclodextrin, and cyclodextrin has a cavity that can capture cholesterol or fluorescent dye. When fluorescent dye is there, fluorescence is quenched because of graphene, and when cholesterol replaces this organic dye, fluorescence is on. This can be used for cholesterol detection. Magnetic separation of protein. Magnetic nanoparticle will functionalize with some molecules so that it can bind with a protein which is histidines. Histidine-rich proteins can be captured by these magnetic nanoparticles. And once you have a magnet, you can isolate this protein from a complex biological environment, and you can further purify by releasing this protein. Colloidal mesoporous silica. Colloidal silica is a small dots are actually iron oxide. Iron oxide is inside. So this leads to magnetic colloidal silica. And silica is very porous. And because they are porous, they are a lot of cyclodextrins are attached at the surface. So this can be used for magnetic separation of cholesterol, much more efficient separation of cholesterol, because this large surface area will lead to many attachment of cyclodextrins, and more amount of cholesterol can be bound to these systems. So a lot of fluorescent dyes are used as probes for imaging different subcellular compartments. These are standard biology labs. They use a lot of reagents, fluorescent reagents for tracking and imaging different subcellular compartments. And this is very important to understand different features of diseases that are happening at the early stage inside cells. So can we replace all these dyes by nanoparticles? Why do we need it? Why do we need to replace dyes? Here is the reason. So the cell, living cell, two rows, top rows, bottom rows. Top row you see cytoskeleton is labeled with organic dye, that's the conventional one, and the nucleus is labeled with quantum dot, inorganic, the nanoprobes. And with time, in the sort that 0 second, 20 second, 60 second, 120 second, the exposure time. We are exposed because we want to see what is happening. We have to expose the sample cells with the light, and during exposure, you can see the problem of organic dye. Organic dye bleaches, fluorescence finishes, but for quantum dots, it don't bleach. So this is a common problem of organic dye. The bottom rows are again same, just reverse. Quantum dot is used to label cytoskeleton, and organic dye is used to label nucleus. You can see the organic dye bleaches, nucleus vanishes. You don't see nucleus after some time. So common problem of organic dye is bleaching. So this bleaching limits application. If we want to say, for example, single molecule imaging, small nanoscale imaging, continuous imaging, video imaging, these are not possible with fluorescent dyes, very limited. So quantum dot can be used, fluorescent nanoparticle can be used, the replacement. How we do it? So culture cells, then our nanoprobe solution, add it in the colloidal form as a food to the cells, because cell culture means you need to supply foods in an aqueous solution. So there you can mix your nanoprobe. And after that, you need to wash, and label cells can be imaged under microscope, and many other flow cytometry and other things can be done. So we can design wide variety of nanoparticles, nanoprobes, different size, different surface chemistry, and depending on their surface chemistry and size, different uptake mechanism, different processings. And what we do with them? You can see the labeling of different compartments: Golgi, cytosol, MTOC, lysosome, mitochondria, lipid droplet, perinucleus, nucleus. So many compartments if you want to label. And not only that, you can clear some of these toxic materials from the cells. You can follow the drug's activity inside cells. And again, I will quickly go through some of the examples. So top rows is actually same materials, same quantum dots, changed surface chemistry. By changing surface chemistry, the bottom row you can see their interaction with cells. Bright fields are cells merged with green color fluorescence coming from quantum dot. So green color indicates the labeling natures. Some cases they are only in the membrane, cell membrane. Some cases they enter inside cell. In anionic, they don't interact at all washed away. So by changing surface chemistry, you can control interaction with cells, entry into cells. Again, what I showed, that same nanoparticle quantum dot number of folate is less or high, you can see they are trafficking into cells. Blue one is actually nucleus. And compared to nucleus, and red one is quantum dot. Compared to nucleus, in one cases, less folate means they are in one side of the nucleus. That's actually either Golgi apparatus or something like that. But in other cases, they are everywhere. They are actually, we know that they are trapped in lysosomes, small red dots. A similar right side, another tagged peptide, less and less means they enter into cell and stay at a perinuclear region one side, but a large number they enter and then exit exocytosis. Similarly, here gold nanorod, which has photothermal property. If you excite gold nanorod with light, they capture visible light and translate into heat, and that can be used to kill cancer cells. So gold nanorod here is functionalized with some antibody and then label cancer cells and then shine light to kill cancer cells. Here are the examples. Different nanoparticles to label different subcellular compartments. Tumor labeling. Here actually gold nanoparticle and then functionalized and loaded with some Raman probes and then injected in blood tail vein of a tumor mouse, and then collected tissue samples from different parts of this via biopsy, and then used surface enhanced Raman because probes are there in nanoparticles, Raman probes, organic dyes. Their vibrational finger can be used to trace where are those nanoparticles, either in tumor or somewhere else. Why do we need this? We can prove that whether they are targeting to tumors. If they target, we can use drugs to deliver on those organs, tumors. Cancer cells, when they are in advanced stage, they produce MMP, matrix metalloprotein, so that cell can migrate from one side to other and tumor can spread. And medical surgeon requires, during the surgery, they need to see which part of this component of the area they should remove. And for that, ideally they need an imaging-based approach to see which area they should remove. So here, people design quantum dot functionalized with some peptides, which can be used to label that cancer cells which are in advanced stage, which are about to migrate, which are migrating. So this enzyme can switch on the quantum dot probe, otherwise they are off. Just a second. Hello? কবেকার কথা? আরে আমি এখন ক্লাসে আছি, একটু পরে। So MMP can be used to MMP is an enzyme that cancer cell produces that can be used to switch on these quantum dot fluorescence. Quantum dot fluorescence are off because they are linked with some certain dye that quenches this fluorescence. Once the MMP is there, it keeps this peptide bond, fluorescence switch on, and they enter into cells, make them cationic, and the nanoparticles highly enter into cells, and that's how fluorescence switch on. And this is a designed nanoparticle, this micelle, polymer micelles, that can be used as a delivery carrier for DNA or macromolecules, proteins also. So this is here, it is showing that DNA can be delivered. The DNA delivery is very important. cell delivery of DNA because they are kind of there are many kind of genes that need to be delivered into cells for that they sometimes act as silencers, gene silencing, or sometimes they can promote certain specific protein overexpressions. So for that you need to deliver genes into cells. So this is a carrier, nano carrier that bind at the surface of the gene can bind at the surface and deliver by translocation of the cell membranes. These are a few more examples. These two different drugs are loaded into nanoparticles. It's a polymer nanoparticle. Two different drugs. One drug is inside nanoparticles and covalently attached. Another drug is outside, that is weakly bound. The first drug that is bound outside, that can easily come out. That comes within a few hours of injection into a body. And these nanoparticles will travel through blood, go to the tumor site, and that area, first drug will be released. And this drug will stop. And this is anti-angiogenesis drug that will stop blood vessel formation, block these blood vessels, so that cancer cells that like to migrate, they cannot migrate. The first drug action will start. And then the inner drugs, they will come slowly. That will enter into cancer cells, enter into the nucleus of cancer cell and block the cell replication, divisions. So these drugs, these nano drugs are much more efficient than conventional single drugs. We can make nanoparticles as a delivery carrier or delivering proteins into cells, cell nucleus. You can also make drugs that can be used to image tumors in animal models. We can also image right side imaging mitochondria in a cells. Why do we need to image mitochondria in a cells? Because mitochondria's function, this function is linked to many different diseases. So early stage disease, mitochondria functions are linked. Glucose responsive delivery. This is important for insulin delivery, for example. When you have high blood glucose, you need to deliver insulin. When low blood glucose, you don't need. So you need glucose responsive delivery of drugs. There are many reasons for glucose responsive delivery. So this is a material where this is a porous material, and they have a lot of windows or gates. These gates are blocked. And inside, drug is inside. So each mesoporous materials loaded with drugs, but their windows or gates are blocked. These gates can be opened by glucose. It's a dextran-based polymer that are used to block these windows or gates, and in presence of high glucose, they will be removed and the drug will be released. Glucose responsive delivery. Vitamin C, they are antioxidant, but vitamin C we cannot make, we have to eat every day. And if we eat too much, that will our body cannot take it. So small amount every day, that's important. So vitamin C antioxidant, but it can, if you use high dose, they can act as a pro-oxidant. This is the chemistry involved here. And so delivering vitamin C into cells in a high dose is important. If we can deliver vitamin C to cancer cells, they can kill cancer cells. So here we can design nanoparticles so that they can target cancer cell, and in presence of vitamin C, they can produce reactive oxygen species, and through which they can kill the cancer cells. This is another NF.TiO2. You can spray them in the fruit which are infected. But you have to have visible light. In presence of visible light, these nanoparticles that are sprayed, they capture visible light, make reactive oxygen species, and they can kill these fungus. So fungal therapy is possible. And many different nanoparticles which can produce reactive oxygen species. Once we can deliver them into cells, they can be used to control cellular functions, autophagy. Autophagy is a cellular self-eating process, and this process is regulated for controlling diseases. These are involved in different kind of diseases. Up-regulation of autophagy is linked to certain diseases. Down-regulation of autophagy is also linked to certain diseases. And by using nanoparticles that can produce reactive oxygen species, control autophagy, we can actually use them for therapy. Cell therapy for controlling disease therapy. Sound can also be used for therapy. We know we can hear only a certain range of sounds, and ultrasound can be used for therapeutic application. And ultrasound can be used as a pressure, mechanical pressure that can be used to do surface electrochemical reaction at the surface of piezoelectric materials, pressure sensitive materials. Piezoelectric materials, in presence of pressure, they can make local charges, and charges can lead to electrochemical reactions. Sound based, and in addition to that, sound can make local heating, cavitation effects leads to rapid the formation of different bubble generation, degradation, collapse. That can also lead to different kind of processes. Processes means chemical processes, and this can be used for different therapy. We can use sound for temporary loosening of cell membranes for delivering drugs, temporary disruption of blood-brain barrier for delivering drugs into cells, biofilm that are produced in a body or in a transplanted organ, transplanted systems in a body. You can use ultrasound-based wireless therapy, cell stimulation, neuron cell stimulation based therapy, and degradation of proteins which are linked to Alzheimer's. Okay, maybe another 10 minutes. So protein, as I said, protein aggregation is linked to different neurodegenerative diseases, and in cases of Huntington, you know that this is linked to, it's a genetic disease, and it's linked to formation of certain proteins which are called polyglutamine. A protein which is continuous more than 36 glutamine at a stretch, and these proteins aggregates in a neuron cells and disturb neuron functions. And similarly, other cases, Alzheimer's protein, they also have some fibrils that I showed. So these protein aggregations, we now know that there are many chemicals, biochemicals, that can inhibit this protein aggregation. These are the least. And delivering these proteins into cells or body is very challenging, because most of them, you see, they are insoluble. And in some cases, they are very unstable, chemically unstable. Vitamin, EGCG, the first one, EGCG, that is present in green tea. So they are very unstable. How to stabilize, how to use them effectively in bodies for Alzheimer's or Huntington disease. We can make their nanoparticle forms so that it can stop their aggregation, it can enhance their stability, it can enhance bioavailability, enhance their cell uptakes. So nanoform can enhance up to 100,000 times enhancement in their performance. Performance means how to make, again, similar to chemistry processes, and how to see whether they work well. These are the model systems. We can make in laboratory different fibrils in standard conditions, and in presence of our molecules or nano drugs that we design, we can check whether these aggregations are stopped. We can have animal models to see whether they can be stopped in mouse models. And in the cell models, we can actually use microscope, fluorescence microscope, to see aggregations. The two images in left one is actually the mutant Huntington protein that are expressed in cell. They are in the green color. Green fluorescence is coming from proteins which are tagged with GFP, green fluorescent protein. So because of this green fluorescent protein, you can see proteins overexpressed, but they are everywhere. They are distributed in cytosols. But the right side, they're aggregated, dotted structures. These dotted structures means proteins are aggregated. Cells are going to die. So by following microscopy, fluorescence microscopy, you can see the stages of cells, whether cells are going to die. And by using this microscopy, we can evaluate our nano drugs. Also, you can use electron microscope. The small dots are actually our nanoparticles, and the fibril structures are protein fibrils. Left side one, see, nano drugs, nanoparticles are attached in the fibrils. The right sides, fibrillation is inhibited. That's why you don't see much fibrils. At the nucleation stage of fibrils, nanoparticles are attached and stop the fibrillations. As I showed, these nanoparticles that can be used to inhibit protein aggregations. So in the middle, you can see two cell images. The top one, aggregated proteins in the form of green dots, fluorescent green dots, proteins aggregates. Here nanoparticles are not used, our nano drugs are not treated, that's why untreated. But the bottom one, cells are treated with nano drugs, nanoglutamine. You can see there is missing fluorescent aggregates, which means protein aggregation is stopped. You can also do Western blot and other protein analysis that is shown in the right sides. So why nanoforms are important? Why nanoforms are important? Because nanoforms have three advantages. One, if they are organic molecules you want to deliver into cells, you cannot do it because organic molecules are water insoluble. So you can, in the nanoform, you can disperse them in water. Second advantage is they can have very high cell uptake via endocytosis. And third one, you can stabilize them. Some cases, if they are unstable, chemically unstable, you can stabilize them. Self-cleaning surface. Now there are people are making paints which are self-cleaning. If you paint them in the wall or in your shirt, you don't need to clean the surface again and again. In order to have that, you need to have the lotus effect. What is that? So you need to have a micro scale features, micro nanoscale features in your surface, in your materials. So you have to design them. Again, you have to make nanoscale materials, nanoscale designs. For environmental pollution, also you need nanotechnology. You can make particles like your green plant, how they use sunlight to make foods. Similar approach you can use, capture visible light and then use them to degrade materials, toxic materials, infect fungus. For that you need to know this second kind of contaminants called endocrine disrupting chemicals, mainly toxic chemicals and biochemicals, which we need to be very careful. This is a new generation of toxic materials. People are more scared of it, and people are doing research on this. These chemicals do a lot of harmful effect in our body. The reason they are endocrine disruptor, they disrupt our endocrine systems. They mimic our endocrines, some of the hormones which are involved to the endocrine systems, stimulate our endocrine systems, because they mimic in a small amount, micro concentration, micromolar, nanomolar concentration, they are very toxic. So their removal from the foods, from the groundwater, mineral water, drinking water is very challenging aspects. So we can design nanoscale materials, functional nanomaterials for the separation, for the degradations. These are some of the examples. Some of the functional materials, magnetic materials, colloidal materials can be used for separations. Magnetic separation, magnetic graphenes, separation, and graphene-based silver or gold composites for sunlight-based degradation. So you need to make large scales for applications, real life applications. And we also can design materials for ultrasound-based water purification. For example, in your columns, water column that you use for separations, you can fill with your sound responsive materials like nanoparticles made of piezoelectric materials, barium titanate, for example, and use ultrasound for purification, degradation, as well as disinfection of bacterial disinfections. You can use these kind of materials. These are mostly in the research stages. I think I should end. So what we understand from this talk, we need to understand the property of materials, property of nanoparticles, size dependent property. In particular, we need to know what materials make nanoscale size dependent properties. We have to identify, and then we need to use chemistry to make nanoparticles of right size. We need to make functional nanoparticles, and then use these properties of these nanoparticles, either core or functional properties, for exploiting them, for these properties, exploit their properties, for use them as a medical imaging probe, bioimaging probes, or use those properties for therapeutic applications, or use them for environmental remedy. So it's a vast area of research, a vast area, field is very vast, and it needs understanding of biophysics, biochemistry, and in order to make materials, you need to understand chemistries, and even many materials are commercially available. You need to understand what chemistry, what kind of applications you need to perform. For that, you need to use right chemistry, choose right chemistry, choose right product, select right product for their applications. And also, in terms of instrumentation, also you need to have instruments like, for example, fluorescence is most common instrument for the application of some of these nanoprobes. But in addition to that, gold and silver, you can also use other microscope like dark field microscopies. And for the therapy applications, you need to know what kind of therapy. It may be photochemical approach, it may be sound-based approach, it may be selective. And for the biomedical application, you need to know what well-defined biological problems where you want to solve. I think I came to the end. Thank you for your patience. And if you have any questions, you can ask.
Thank you, sir. Thank you, sir. Thank you, sir,
yes.
for this beautiful presentation on biomedical application of nanoparticle. The session was very informative. Now we will move to our question answer session. I will request to our teacher participant if they have any question, they can ask one by one.
Sir, may I ask you one question?
Mm-hmm.
Sir, you told you have shown in one of your slides that nitrogen sulfur dope TiO2 is being treated was being treated into fruits for removal of fungal infection. So my question is, how could we measure how much nanoparticles is needed to stop the fungal infection? And the second one is whether it is toxic or non-toxic, whether the consumed nanoparticles is going to our food system. I mean, we are eating the raw
Yes.
foods also. So is this good?
Yes. Very good question. First of all, you understand this is a research stage, and these are spread into the surface of this fruit, and you can expect some might be entering into the cells into the fruit. Their chances are less, but we tested their cytotoxicity. TiO2 is used as a paint. It's a huge amount of environment. Exposed in the environment, so they are less toxic in terms of they don't kill cells, but where in presence of light they can kill cells, because in presence of light they make these toxic chemicals. So generally toxicity is less, but if you eat too much, there might be a consequence of toxicity that we have not tested. And people, this is a research area where people are working. A lot of people are working toxicity of nano things. But if you want to use it, you have to use in the spray small minimum amount that will work.
Okay, sir. Sir, I have another question. You just showed that you have explained about the what's called fibrils. So my question is the amyloid, beta amyloid fibrils, lysosome fibrils, insulin fibrils. So how could you check the selectivity that whatever the nanofibrils are nanoparticles are producing and adapt with different biofibrils. So how could you check the selectivity and how could you design to make it selective? I mean, the quantum dot is particularly selective for amyloid beta or lysosome or insulin. It is been tested in vitro process or in another way. Otherwise, it can attach in any of the biomolecules.
Yes. Yes, very good. Okay, I hope you understand the subject. That's good. Actually, the whole process of this functionalization is actually to make them selective, so that it can interact with only the system that we want. For the fibrils, say people, just nanoparticle will not work. You have to first coat with something. For example, I showed that polyethylene glycol kind of molecules, so that they stop interacting with anything. And then you attach specific molecules. For example, in your peptide, the fibrils, you can attach some specific peptides which are known. They interact because this is a small component of the fibril forming molecules, fibril forming proteins. So that peptide we know. So that sequence you have to attach on the nanoparticle surface. That will lead to binding with your fibrils. And there are certain molecules, small molecules, they also interact with the proteins. They via this they enter into the beta 6 structures, hydrophobic components. For example, some dyes, thioflavin D, we know that they bind in the hydrophobic pockets of the fibrils. So we use those kind of binding interactions for selective interaction with the fibrils. So this you have to test, and these testings are possible. Like I showed one TA micrograph where fibrils are bind with the nanoparticles. You can clearly see, and these are the options for testings. In addition to microscopy, you can also see by the spectroscopic ways like fibrils precipitate. Your nanoparticles has a color and they are colloidal, they don't precipitate. If your fibrils mix with your nanoparticle, if they bind, then fibril will be color, and you will see color precipitate. These are indirect way of observing interactions. There are many ways of monitoring these interactions.
Okay, sir. Thank you, sir. Thank you for your nice lecture and lecture also.
Okay, any other questions?
Sir, thank you for the excellent talk. I have a question regarding the colloidal nanobioconjugation onto that amine particle, aminated particle. So you mentioned that we can conjugate like using NHS, right? So I have seen like many times people have also used NHS, sulfur NHS.